Hi everyone. Today we would like to discuss about the DNA replication. Before that, we have to introduce ourselves. My name is Noraini binti Mustafa. My matrix number is 206669.
My name is Eng Yi En, and my metric number is 205,828.
My name is Eng Jiami, metric number 206504.
My name is Nafagira binti Ahmad Kamal and my matrix number is 205275.
My name is Nahan Nizam binti Mersid and my matrix number is 205477.
Okay, so we start our discussion now. Okay, DNA replication is a semi-conservative process which a parental strain is used to synthesize a new complementary daughter strain using several proteins, which include enzyme and RNA polymer. But before that, the DNA strain is belong to what organism? Is anyone have any idea?
It belongs to eukaryotic organism because it has many origin of replication. If prokaryotic organism, it only has one origin of replication, so the DNA strands belong to the eukaryotic organism.
I agree with you, Jamin. Um, by looking at this figure, there are two origin of replication and the DNA also a linear. So DNA replication have three steps or stages which are initiation, elongation and termination. So what really happens to DNA strain at the initiation stage? Is anyone want to try?
Before that, I want to add something. Why replication must be fully methylated before initiation occurs?
I would like to try. Is it for eukaryote also want to methylate it at the origin of replication?
I think yes.
If if based on my understanding, if for prokaryote we need to methylate because if for prokaryote but I'm not sure whether it also apply for eukaryote or not. If for if for prokaryote the it do methylation at the origin of replication because for prokaryote the bacteria is able to differentiate between the old strand and the newly synthesized strand. If first before replication the origin of replication is fully methylate, like both parental strand have the methyl group at the adenine side. So after it do the replication, it got the hemimethylate, like only the parental strand got methyl group at the adenine and the daughter strand doesn't have. By this, bacteria can do like proofreading, like find whether the synthesis, which is the daughter strand, which is recognized by the non-methylate adenine group. If they find there is a missed pair of got something wrong with the synthesis daughter strand, it will recruit the repair enzyme and come to repair it. This is for the prokaryote, but for the eukaryote, I'm not sure whether the mechanism is same as the prokaryote or not. Mm.
Is it clear for you, Fagira?
Yes. Thank
Okay.
you.
Uh, is anyone have a question?
No.
Okay. For initiation stages, I will let you try. Okay. For initiation stages, DNA synthesis is initiated within the template strain at a specific coding region, which is the origin, and the origin site are targeted by the initiator protein, which are DNA A protein, which recruit additional protein that help in the replication to form a replication complex around the DNA origin. Then there are several origin sites on the DNA strain, which known as replication forks. The replication complex contain the DNA helicase that used to unwind the double helix, exposing the two strain, which to act as the template for the replication to occur. And then the mechanism of DNA helicase is hydrolyzing the ATP to that used to form the bond between the nucleotide, thus breaking the bond between the two strings. And then during the initiation of DNA, DNA primase synthesize small RNA primer that kickstart the function of DNA polymerase. That's all for me. Is anyone have any question?
Yes, I want to add something. You mentioned that DNA helicase separate the double strand. So once the single-stranded DNA is formed, the single-stranded binding protein will bind to the single-strand DNA because binding of single-strand binding protein will stabilize the DNA so that the DNA will not self-annul or hydrolyze by the nucleus. Once it binds to the single-strand DNA, it will promote the binding of another single-strand binding protein. And then this interaction will stabilize DNA. So once the DNA stabilizes, then it can proceed to the next step, which is binding of RNA primer, then elongation, then termination. That's all from me.
I would like to ask one question. Is it for the origin of replication for the prokaryote, the conserves, like the consensus sequence is same for the eukaryote? Like for the prokaryote got the um, 4 copies of 9 base pair repeat. Is it same for eukaryote also have this kind of copies?
I think different because prokaryote only have one origin of replication, but in eukaryote they have many origin of replication. So based on this, I think quite different.
Okay,
Because
thank
in
you.
because in
Okay, okay.
eukaryote, replication can happen at um, different different part because it has many origin of replication.
Hmm. Okay, thank you. I would like to add something for the initiation process. For the enzyme helicase, it actually attached to it has the polarity it moved from polarity from 5 prime to 3 prime. So it actually attacked at the lagging strand. Mm.
I want to ask, I don't know, ask, eh, just now, Ian just said that, fully ventilated origin only exists at prokaryotic and you are not so sure at eukaryotic. But here, as we know, uh, only fully ventilated origins of replication, it will uh, active the origin and ready for the replication. Or and if at the eukaryotic, just say lah, just say we don't have we don't need it fully methylated. So is it it's still active the origin and ready for replication, or it has the other ways? Do you understand what I mean?
Yes, I understand. But I try to Google search.
Is Haniza here? Oh, he just she just left the meeting.
Fadira, can you repeat your question? I quite blur.
Okay. Earlier you say uh only fully methylated will exist at prokaryotic, and she not so sure about eukaryotic. And my question is, uh, if if in eukaryotic doesn't have the fully methylated before initiation occur, how does the replication begin at eukaryotic? Uh,
Okay, thank you.
okay.
I think for this question, we need to search for some answer. Is it okay for us to proceed to the next one? Then after we finish our discussion, then we answer the question.
I think it's
Yes,
good
yes,
idea.
sure. Okay.
So we is anyone have any question or want to add something? No. Is Haniza okay?
Okay.
Okay. So we proceed to next. Okay. For the alien question, state what really happened to DNA strain in the initiation state? Is anyone have want to answer this question?
I would like to try
Okay.
the initiation at the is it the elongation?
For elongation.
Based on my understanding, the elongation is marked by the first phosphodiester bond that formed between the terminal hydroxyl group of the RNA primer and between the deoxyribonucleoside 5' triphosphate, which the hydroxyl group will act as the nucleophile and attack the alpha phosphorus atom and form the first phosphodiester bond. Then after that, the DNA polymerase will come to not uh, this is doing by the DNA polymerase 3. Then it continue to synthesis the DNA strand by using DNA template. And for we know that the replication for eukaryote is replication is semi-conservative, and in eukaryote it have the leading strand and the lagging strand. For leading strand is continuous, and for lagging strand is discontinuous. So overall, we known as semi-discontinuous. And then for the um, does anyone would like to continue?
I want to add
Can I
something.
try?
Oh, Hani, okay, okay, Anissa. Is Anissa okay? I think you are quite lagging.
Uh, I will give Fahira to explain the elongation.
Okay. I just want to add a little bit. In elongation, it has DNA polymerase 1 and DNA polymerase 3, but in elongation, DNA polymerase 3 more suitable because it has the higher accuracy and can edit the ATP more effective and more faster. Like DNA polymerase 3 can add 1000 ATP per second. Okay, Anissa, you can continue.
Haniza, you lagging?
Uh, okay, okay. Now, okay. Um,
Okay.
Is it still lagging?
I think Haniza have left the meeting.
Or if like that, then I continue first.
Uh,
Okay. Haniza,
yes. Um, does anybody want to add more for elongation? Because I want to proceed with termination.
Yes, I want to add something with elongation. For the leading strand, DNA polymerase chain involvement in lagging strand other than DNA polymerase 3, DNA polymerase 1 also involved because polymerase 1 is help to fill in the gap. So to make it simple, because lagging strands have many fragments, which is also known as Okazaki fragment. Between Okazaki fragment, there is a gap. The gap need to be filled in so that a complete daughter strands can be produced. Not used to, while the polymerase one is work quite slow compared to polymerase 3. If the gap between lagging strands is have any mistake, then the polymerase 3 cannot reverse back and repair. While for polymerase 1, if the gap between lagging strands is have any mistake, then the polymerase 3 cannot reverse back and repair. Process is it carries the process quite slow to add.
I would like to add something. For the DNA polymerase tree, it more prefer to synthesize the DNA from 5 prime to 3 prime. And then I'm quite confused. Just now you say for the DNA polymerase tree cannot repair it. Is it? Oh, I miss here.
What I mean is reverse and repair. It can repair, but it cannot reverse and repair.
Oh, cannot reverse and repair. Um, if the DNA
Okay.
polymerase tree have the 3 prime to 5 prime exonucleases, is it still cannot reverse and repair?
Um, Sorry to interrupt, but I think we should end this meeting first and continue.
Okay.
Okay.
Okay. You can start.
Um, okay, I would like to add just now, Jamie, you mentioned that the DNA polymerase tree cannot reverse and repair. Then, but it have the 3 to 5 prime exonucleases. Is it still can reverse to repair?
So if it has 3 prime to 5 prime exonuclease, it can reverse. Is it because I quite confused?
Okay, so it can reverse and repair. Okay, then I would like to add something. The reason why the DNA polymerase 3 is more suitable than DNA polymerase 1 to do the DNA replication is also because of the processivity. Processivity, which is the like number of the deoxyribonucleotide triphosphate that the enzyme can fill in the template strand before it dissociates from the template. And for the DNA polymerase 3, it has high processivity, which is up to 50,000 like that. And for the DNA polymerase 1, I have low processivity. And DNA polymerase 3 also have high fidelity and high accuracy, like Fagira mentioned just now. Mm. Does anyone like to add something?
For me, no.
I want to add something about the exon, the replication for stop or slow down, which is cut off remove the phosphodiesterase bond. So if isonuclease cannot function well. Other than that, proofreading only can be carried out if the speed is slowed down or error is detected. So I think that's why, but yeah, nothing. That's all from me.
Mm, I would like to add for the DNA polymer 3 and 1. For DNA polymer 3, it only have like 5 to 3 prime polymerase and 3 to 5 prime exonucleases. While for the DNA polymers 1, it have 3 domain, which is the 5 to 3 prime polymerase domain and 3 to 5 prime exonucleases domain and 5 to 3 prime exonucleases domain. So because of this, the DNA polymerase 1 will tend to remove the RNA primer at the lagging strand because the RNA primer is synthesized from the 5 to 3 prime direction, right? So the DNA polymerase 1 will use its 5 to 3 prime exonucleases to to remove the RNA primer and use it 5 to 3 prime polymerase to fill the to replace the RNA primer by using deoxyribonucleoside triphosphate. Mm.
Is anyone want to add something?
For now, no.
Okay. So we proceed to termination. Um, is there anyone want to explain about termination stage?
Uh, okay, I will try.
Okay.
In termination, the NA polymerase will remove the RNA primer from the lagging strand, and then it will replace um, with DNA with nucleotides. After that, DNA ligase will combine two nucleotides from the Okazaki fragment by forming phosphodiester bond between the 3' OH of one nucleotide and 5' phosphate of one nucleotide. Um, I think that's all for me. Anyone want to add?
I want to add about termination, which termination occurs when two replication forks meet on the same strand of DNA, as Anissa said. Then termination is different in eukaryotes and prokaryotes, which in prokaryotes, after replication complete, daughter DNA molecules will remain linked and type 2 topoisomerase will cut one of the DNA molecules and will reduce it. Then for eukaryotes, there is no sufficient base for the primase to synthesize RNA primer at the end of the chromosome in lagging strand DNA, which there is a small gap left at the end of the chromosome. Then the small then the special region then the special region at the end of the chromosome we call as telomeres. That's all for me.
Mm, I want to add something. For termination, the telomeres are synthesized by a special type of DNA polymerase enzyme known as telomerase. Telomerase can catalyze the telomere sequence at the end of the DNA sequence. And on the completion, the parent and complementary strand coil into the double helix shape and producing two DNA molecule, and each DNA molecule passing on strand from the parent molecule and one new strand. I think that's all for me. Is anyone want to add something?
I would like to add something. For the telomerase, it have the RNA template, which is used to, like just now you say, synthesis to expand the DNA template, which is short, and the RNA primer cannot have sufficient place to synthesize the primer, right? So it use its RNA template to synthesize the DNA and allow the primaries to make a RNA primer, and for the DNA polymerase 3 to continue its synthesis it to continue synthesis DNA. And then for our human somatic cell, we have low telomerase gene to synthesis the telomeres. So our so after cell division, if the repeat sequence of the telomere will continually become shorter. And if it's shorter until affect our important gene, it will cause abnormality of the cell. And for the cancer cell, it have like many telomeres. So it cause it can sort of like replicate many times and many more and cause the tumor. This is just based on my understanding. Does anyone would like to add something?
I have question. Does telomerase exist in all eukaryotic cells or just certain cell?
All eukaryotic cell. Because most eukaryotic cell have linear chromosome. If circular chromosome, that's a different thing, which is belong to prokaryote.
Okay, thank you.
I want to ask something. Why is that the cancer cell have a lot of telomere? Like Ian say just now.
I think but I'm not so sure. Just I think because the cancer cell it need to proliferate very rapidly and many times and cause a tumor. And because it can proliferate if it has the sufficient telomere gene, then it will provide many telomeres.
That's all my I think that is because of mutation. Maybe there is some mutation in the telomeres and then cause it to increase its amount exponentially, and then cancer cell have produced.
I agree with you.
Okay, understand. Is there anyone want to add something?
Now from me.
Ian, do you have a question or want to add
I
something?
think because based on the question, we can see got two replication for I. So I think after the end of replication, they will fuse together because it got two replication for. Is it will fuse together?
What I think is same with you. Maybe they will fuse together.
Hmm.
Fagira, do you have a question or want to add something?
No.
Okay. Haniza, do you have a question or want to add something?
No.
Okay. I think that's all from our discussion. Thank you, everyone.
Thank you.